Our last two groups of molecules are proteins and DNA. And I like having them in the same video because they are actually very much related to one another. So, sort of some context for where we are going with our discussion of proteins and DNA, and as sort of a preview for a couple of our chapters later. The structure of our DNA is such that it is information coding, which means you can use the sequence of the DNA monomers called nucleotides to produce proteins. So in this image here, we have a DNA sequence, and that sequence is read by a molecule that is a protein and used as a coding sequence to string together protein monomers. So the function of our DNA is to basically be the blueprints or the directions for producing proteins. Now this process is called gene expression. And we're going to spend a couple of chapters talking about how DNA works as a coding molecule and how it is used to make a protein, which is going to have a functional purpose within our cells. Now, again, going back to the structure and function thing, this picture here, this diagram here, is showing what happens when you change the structure of your DNA. So changing one of the molecules in DNA is called a mutation. And by making a mutation in the DNA, we change our DNA sequence, which changes the code that can be read by our protein building molecules. And those protein building molecules are then going to build a different protein. So our example here is sickle cell or sickle cell disease. There's a couple different representations of it. On the left-hand side, we have the change in DNA sequence, which causes a change in your protein sequence. And it causes a structural change in the proteins that are being produced. So we go from having this nice, beautiful portion of a hemoglobin molecule to this one with this little divot in it. Now that affects how they're put together into a hemoglobin molecule. The purpose of hemoglobin is to carry oxygen, and when the DNA sequence is changed, which causes a change in the structure of the protein, the function of that protein changes. It can no longer carry oxygen like nice, neat, normal hemoglobin. The reason that it causes problems in the cell is that, one, it doesn't carry oxygen as well, but two, it produces, instead of these nice, round, beautiful blood cells, these sickle or crescent shaped cells. Those cells don't carry oxygen very well. They get clogged in veins and arteries. They cause all kinds of problems for individuals with the disease. And we'll talk about sickle cell disease a little bit later, but I wanted to show you how our DNA structure affects our protein structure, which then affects the function. So let's talk about proteins and how we build proteins. We'll talk about how DNA is actually used to build proteins in a unit at the end of the semester. So we're not going to worry about how these proteins get built yet, but we are going to worry about what they are made of. So again, you all know what proteins are. Proteins generally come from eating animals, so fish, beef, chicken, eggs, things like that. But you can also get proteins from things like nuts and seeds. Soy also has protein. There are some plant-based proteins that are really, really healthy for you. So proteins are important because at the molecular level, proteins govern, mediate, perform nearly every function in a living cell. I like to say that proteins are the worker bees of the cell. They are the ones that are mediating and performing all of the metabolic processes. They're making things move. They're fighting off infection. They're really, really important. So important, in fact, that they make up about 50% of the dry mass of a cell. So if you take a single cell, dry it out like a raisin, 50% of what's left is going to be protein. Proteins have can have really complex and diverse structures, but the basis of a protein structure is very similar to a carbohydrate in that it is just a repeating chain of the same type of molecule over and over again. So the primary structure of protein is really similar and simple amongst all proteins. But the way the actual sequence of the monomers will produce different chemical interactions, which can create these incredibly complex and diverse structures. Those diverse structures means that we get diverse functions, which is why so much of our cell function depends on proteins. So our proteins are made up of amino acids. Proteins are a polymer of amino acids, which means they are a chain of all these repeating units called amino acids. Now, the initial chain of amino acids is unbranched. It looks like pearls on a string. It's just, at least in this picture, it is a repeating structure made up of two parts. We have this bottom part that has an amine group and a carbonyl group, and then we have this other structure that actually differs amongst amino acids called the R group. Now that R group is going to come off of this central carbon. The bottom here, the amine group, hence the name amino acid, is going to be the same. We've got the nitrogen and the carbonyl group. But those R groups are going to change. And those R groups each have specific properties, and that's going to help make our proteins really, really diverse. There are 20 different amino acids. And those amino acids, as I said, differ based on their R group. So this image here shows the amino acid backbone and then the different R groups that are attached to them. Now, the different R groups have different categories or types of properties. We've got some that are nonpolar, which means they're hydrophobic. They're not going to form acids. They're not going to interact well with water. They might interact well with each other, but they're not going to interact well with water or other polar molecules. And then we've got the polar or hydrophilic R groups. Those are going to interact well with water. So you might already be thinking, well, if you have nonpolar groups and you have polar groups, different regions of proteins can actually behave in different ways. And that's really important. And we're going to talk about that again in chapter 5 when we talk about our membrane structure around our cells. We can also have acidic or basic R groups. The acidic ones are going to release hydrogen. Remember, our acids in chapter 2 release hydrogens into a solution. And our bases either release a hydroxyl, an OH group, or they accept a hydrogen or reduce the hydrogen ion concentration. So of course, those are going to give amino acids specific properties as well. So all of these amino acids are linked together in a specific chain, linked at their their amino acid backbone are linked together with those R groups coming off. just like our other macromolecules, amino acids are produced, polypeptides or amino acid chains are produced by dehydration reactions. So again, we are going to pull a hydrogen and a hydroxyl group off of our monomers, release water, and link them together. So here we have our amino acid backbone and our R groups coming off. You can see that this chain is starting to form here. Now the bond forms between the carbonyl group on one end, carbonyl group on one end and amino on the other. Our proteins have a direction. So one end is called the N terminus because it's got an amino acid. At the end of the chain, at the other end, we've got our C terminus, our carbon end, and there's a carboxyl group at that end, the C double bond O to OH. So we say that our polypeptides run from N to C, from our N terminus to our carboxyl group. So you guessed it, structure and function is really important for proteins as well. And the structure of a protein is going to be based upon the amino acid sequence. In order to get the final structure of our protein, in order to get our final function out of our protein, the polypeptide chain has to be folded. So the structure of that amino acid sequence, the R groups in the amino acid sequence, are going to determine how the protein folds and how the protein works. And that structure is going to give each protein its own unique function. So we have a couple examples here of proteins. One, on the left-hand side, we have a protein from a flu virus. So this is possibly a protein on the outside of the flu virus, a protein that the flu is making, the flu is making. And that protein in this image is being recognized by an antibody protein. So antibody proteins are found in our bodies. They are produced in response to an infection. And they are produced to recognize whatever it is that is invading our cell. So in this case, the antibody recognizes that protein from the flu virus, and that allows our body to mount a response where the antibodies detect the protein from the flu virus and then they can attack it. The same thing happens with other types of infections. COVID-19 is another such example, and a great example of structure and function. So similar to this example with the flu protein, COVID-19 has this shape that you're probably all really familiar with. It is called the coronavirus because it kind of looks like a crown. It's got these spikes sticking off of the virus. And you've probably heard a lot about the spike protein. Well, the spike protein is important because its shape, the shape of that spike, fits into something called an ACE2 receptor. Now, ACE2 receptors are on the outside of things like our lung cells, our heart cells, many cells within our body. Now, the point of them is not to bind COVID-19 or another coronavirus, but that coronavirus does fit into that ACE2 receptor. So coronavirus will find an ACE2 receptor, bind to it, and use that as its way to enter into the cell. You don't really need to pay attention to this on the right hand side. That's just a complicated diagram from the paper that I pulled this from. But the other side of this is that our body will also produce antibodies for the coronavirus, for COVID-19, that recognize those spike proteins. So antibodies in our body, either in response to an infection or to immunization will build antibodies that recognize this spike protein, bind to it, and then use that as a signal for our body to destroy it. That is how we mount an immune response to something invading our cells, such as the coronavirus. And it's all related to the shape of those proteins and the ability of those proteins to recognize specific shapes of other proteins. So there are 3 levels of protein structure. And those levels of protein structure are superimposed, which means they build upon one another. Some proteins have a 4th level of structure, and that 4th level of structure really just kind of depends on the lower structures below it and what the protein function actually is. The primary structure of a protein is just our linear chain of amino acids. So this beads on a string here are our cartoon representation of the amino acids. You can see the sequence here in this one. Up here, we see the blow up of this. We've got our N terminus end. We've got our amino acid backbone. And we've got our R groups coming off of this amino acid backbone. Remember that each one of those R groups has specific properties, such as being nonpolar, polar, acid, acidic, or basic. But the primary structure depends on the linking of that backbone. So the primary structure of a protein is just going to be the sequence of amino acids. And as I said a few minutes ago, that sequence is determined by the coded sequence within our DNA. So here we have a long string of an example protein called transthyretin. And we're just seeing here that sequence of amino acids. Our secondary protein structure, the next level of protein structure, is based upon the R groups that are present in that amino acid sequence. Now, secondary protein structure arises from folding of that long chain of amino acids. Now, folding occurs due to interactions between R groups. So if we look here, the red are our R groups. It's hard to say. Red are our R groups, and they are forming interactions with other amino acids. When this happens, we get 2 types of structures that can result. One is called an alpha helix. This is basically a curl or a coil with interactions between R groups causing this coil. Now, these interactions are hydrogen bonds, which means, remember, that they are individually weak but collectively strong. So you get a whole bunch of hydrogen bonds together. That's going to keep these alpha helices in a tight formation. The other type of secondary structure is a beta pleated sheet. Now, this is sort of like a top-down look at the beta pleated sheet. You can see that there are hydrogen bonds between R groups and the other amino acids. If we were looking from the side, it would look something like this. So this is just a different direction. So as an example of a protein with a really unique secondary structure, we have spider silk proteins. So spider silk proteins are proteins that are really strong. They're very elastic. And those properties come from regions of beta-pleated sheets. So this diagram here is showing repeats of protein sequence that produce these beta-pleated sheets. And those beta-pleated sheets are what give spider silk its strong and elastic properties. After our secondary structure comes our tertiary structure. So tertiary structure is folding of alpha helices or beta pleated sheets. And it's made up of all kinds of different interactions between those secondary structures. So this protein image here, this is still transthyretin. You can see in here we've got an alpha helix. These arrows represent beta pleated sheets, so you can see those different regions of structure in the protein. Those different regions are held together into a 3D shape by all kinds of interactions between our groups. So while you have the alpha helices, you might have some other R group interactions that are holding an alpha helix close to a beta pleated sheet, or a beta pleated sheet close to another beta, something like that. Tertiary protein structure is held together by pretty much any type of chemical bond. So if you think back to chapter 2, we had hydrogen bonds, we had covalent bonds. So this interaction here is a covalent bond called a disulfide bridge. It's just a covalent bond between two sulfurs. We have ionic bonds that are really just attractions between different types of charges. Here we have hydrophobic interactions. So these hydrophobic regions are turning towards each other. Van der Waals interactions are kind of similar to hydrogen bonding. They're an attraction between two molecules. So any type of bond that you can think of could be holding together your alpha helix or your beta pleated sheet. But again, these are bonds depending on the R groups, what is actually coming off of that polypeptide backbone. So not only are the R groups important in giving us our sequence, but they're also important in the overall protein structure of these proteins that we're building. All proteins have primary, secondary, and tertiary structure, all of them. Some of them have an additional level of structure called quaternary structure. So there's an example here. Here's still our transthyretin protein. This is made up of 4 tertiary structures put together. So we've got 2 here and then 2 on the back side. On the bottom here we have collagen, which is a structural molecule sort of similar to cellulose in that it is a long fibrous molecule, except that collagen is made out of proteins. So here our secondary structure of each of these is an alpha helix, and then it is the quaternary structure is going to be 3 alpha helices bound together. If you go back, we also saw hemoglobin. Hemoglobin is also a quaternary protein. It's made up of 2 different types of subunits. So hemoglobin subunit A and subunit B, there are 2 of each one. And those are going to make up a single protein unit. So here is our sickle cell example again. I'm really just using this to show you what the quaternary structure looks like. You've got your 2 beta or B subunits, you've got your 2 alpha subunits, and those all come together to make a full hemoglobin molecule. So proteins have a variety of functions. There are 8 main types of protein functions, 8 ways that proteins function in our bodies. The most important are enzymes. So enzymes are basically they are mediating and controlling chemical reactions. So they are, if you're familiar with the term catalyst, they are biological catalysts, which means they are something that make a chemical reaction happen, but they are not consumed by the reaction. They just happen to be biological molecules, because you can also have chemical catalysts as well. But enzymes will help our body perform all of the metabolic functions that it needs to do, all of the breaking down of molecules, all of the building of molecules, all of the making energy, all of the movement. Enzymes are going to help mediate those reactions. A few other types here. We're going to talk about some of these at different points in the semester, so I would like you to be familiar with what the different types are. We've got enzyme proteins, we've got storage proteins. So these are ones that are storing energy or amino acids. In this example here, it's storing amino acids in an egg. We've got hormonal proteins. A great example of this is insulin, which helps mediate our break down our blood sugar. We've got proteins that are involved in contracting and movement, so muscle movement, moving things around the cell. We've got defensive proteins, so these are proteins that are part of our immune system, antibodies that recognize invading molecules and then tag them to break them down or actually do the breaking down. We've got transport proteins. We'll talk about those extensively in chapter 5. They are going to move things across cell membranes, which is really important for getting things into cells and products and waste out. We've got receptor proteins. So I talked about the receptors that recognize endorphins and morphine. Those are proteins that are doing the recognizing and helping to bind the molecules and pass along signals. And then we've got structural proteins. A couple examples there, keratin and collagen. These are both important in mammals, in our hair and nails and skin, things like that. So know these different types, maybe know an example of each one, but definitely the takeaway here is that proteins are really diverse. They have a lot of different functions within the body. So let's talk about enzymes really quickly. I already mentioned they are a biological catalyst, which means they are used to speed up chemical reactions. They're also used to block or regulate them. And by blocking, I mean that they keep them from happening until they're ready. Life would not be possible without enzymes because some of the chemical reactions would go when they're not supposed to, and some would never go without the help of enzymes. So enzymes are really important for regulating the metabolism in our cells. Again, back to protein structure. Protein structure is super important. We've got all of these interactions between our R groups, but sometimes those interactions can be disrupted by the physical or chemical environment. When the structure of a protein is interrupted, we say that it is denatured. So sometimes something like heat or the presence of an acid will affect the structure of the protein by interrupting the bonds that are holding together the the secondary or the tertiary structure. So here we've got this ribbon picture of a protein. The protein whose bonds have been disrupted are called denatured. If your protein is denatured, its structure is messed up, which means its function is messed up. It's not going to perform its function. So denatured proteins are really bad. Sometimes they'll come back together. Sometimes they're permanently disabled in the sense that they can no longer perform their function. If the protein can't form its function in the cell, it's possible that that cell is going to die. When you cook things, you are denaturing. If you cook some meat, you are denaturing the protein. You are breaking apart the bonds in the protein, which is great because that's one step towards breaking down the protein once you ingest it. But it, of course, means it's not performing its function in that living organism anymore. In one of our labs, we are going to talk about or demonstrate how different things affect enzymes and protein function. So different types of physical and chemical things can affect those enzymes. So let's move on to DNA and RNA. I am not going to talk about nucleic acids too much because we're going to come back to them and talk more extensively about the structure when we get to chapter 13, which is our chapter on DNA, and then 14, which is our chapter on RNA and gene expression, because we're all going to need a review of the structure. That's going to come in like week 13 or 14, and I know that you're going to forget the structure by then. So I'm going to review DNA and RNA when we get there. So this is just the basics today. I said you needed to know the monomers that make up each of these molecules. So let's talk about those. The monomers that make up DNA and RNA are nucleotides. Now, nucleotides are made up of three parts. A nucleotide is made up of a sugar. Here's our carbon ring, a phosphate. You can see those phosphates and oxygens up there, and a nitrogenous base. So you might think, hey, that looks a little familiar. We talked about nucleotides when I talked about ATP, because ATP is actually a modified nucleotide. One of the nucleotides is called adenine, and ATP is a modified adenine. It's an adenine that has two extra phosphates on it. Nucleotides are linked together by phospho, which means they're linked together at the phosphate, phosphodiester bond. You don't really need to know what a diester bond means, but you do need to know that the sugar is going to be bound to the phosphate. So here we have what we call the 3' carbon. It just means that it's carbon number 3. The 3' carbon with the hydroxyl group bound to the phosphate group. This is a dehydration reaction. So water is going to be formed and released, and a chemical bond is going to be formed. The overall structure of DNA and RNA, let me go back just a minute, is going to be a linkage between the sugar and phosphate groups, and we call this the sugar-phosphate backbone. So our nucleotides are made up of a sugar, a phosphate, and a nitrogenous base. There are 4 nucleotides depending on which base is attached to that sugar. I'm sorry, there are 5. And they all depend on what the actual structure is. So cytosine, thymine, uracil, these are all single nitrogen carbon rings. Adenine and guanine are double rings. They're called nitrogenous bases because they have lots of nitrogen in them, and they can, I'm going to stop there. Nitrogenous base, because they are rings made up of nitrogen. The whole purpose of DNA is to be genetic information storage. So both our DNA and RNA are going to be made up of linked nucleotides, linked by their sugar and their phosphate groups. And the information in our DNA is coded based on the sequence of nucleotides. So it's going to be a 4-letter code that is going to help code for our protein and some other things in the cell. So DNA is generally our coding molecule. RNA is slightly different. RNA tends to be more of a functional molecule, but it is still made up of 4 nucleotides strung together with a sugar phosphate backbone. It just takes on a different 3D shape than our DNA based on the function that it performs. So there's a whole variety of different RNA shapes. DNA always has the same general shape. DNA is what we call a double helix. Helix meaning twist. So we have 2 strands of DNA nucleotides that form together based on the way the bonds interact and the shape of the bonds. We get this helical shape. So it's sometimes referred to as a twisted ladder, with the nucleotides in the center bound to one another and the backbones on the outside. So the nucleotides kind of make a rung, the rungs of a ladder. And then so we've got this double phosphate backbone that forms a twisted helix shape. Now, we'll talk about the shape a little bit more when we get to chapter 13, but that shape and those sequences are really important for DNA as a coding molecule. This is just a reminder of the link between DNA and protein. So now that we've talked about the shape of DNA and proteins, this might be a little bit more relevant to you. So we've got our DNA sequence of nucleotides: cytosine, adenine, cytosine, guanine, thymine, guanine. Eventually you'll need to know all of those names, but there we have the code. So this structure here is indicating the 2 different backbones of the DNA. We've got our DNA sequence. That DNA is used as a template to string together our individual amino acids to make our functional protein.